Hey, I'm Arne, founder and CEO of AcademicLabs, and in this video, I'm going to show you how you can use AI in our platform to analyze thousands of patents to identify specific insights, and not in hours, weeks, or even longer, but just in a few minutes. So let's first do a search on our platform for, let's say, antibody drug conjugates. So now we search, we see immediately all our data. Let's dive into patents for this example. Let's not pick all the 14,000 patents, but let's go for the recent ones. So let's say the ones that are filed in the last 2 years, 2.5 years. Let's maybe take 2023. And this is across US, Europe, Japan, WIPO. We can do more later on as well. All right, now 1,586 patents. I have a lot of questions. I want to slice and dice the data. So instead of just looking at, okay, who are those organizations with most filed patents in the recent years, we can actually dive much deeper with our AI option. So we're going to go for the second option. We're going to analyze up to 10,000 patents. You can do it yourself in the platform up to 2,000, which we can increase upon request to 10,000. So let's say the second option, and you can see we will use information from the abstracts and claims and get an Excel export file. So how it works, we select all the patents, and then we can ask 20 questions here. I prepared a few questions. I used the AI, the first option in our platform, to come up with a lot of questions where I can ask a question to the most recent 50 publications or projects to identify novel classes of payloads, for example. So here we go. The first question, where does the primary novelty of innovation lie in this ADC patent? Select from the following. So is it new payload, new linker, and so on. So let's add that in here. All right. Second question, identify the target antigen. Answer with the abbreviated name. If nothing is found, print not specified. Also, what kind of cancer is it focusing on? Or not? If nothing specified, say nothing specified. If it's not in the list, answer with a disease following the mesh nomenclature. Then, is the target tumor specific or expressed in normal tissue? Answer it yes or no. So as you can see, we can ask yes or no questions. We can give multiple options. We can ask just open questions, of course. Here we want to know, is it platform? Is it multiple variants of one asset? Is it a single asset? Is that still needed? No, let's skip that one because it's already part of the previous one. Then let's look at the payload. Print the payload, the name of the payload that is used. You can just say print, mention, you can choose the terms. Now, what payload class? What I did here is ChatGPT knows some of the bigger ones, the common ones, but I used our own search into the newest projects, newest grants on ADCs to pick up more novel approaches. Also looked at the latest publications, so with one question, I could quickly pick up some of the more newly established or novel approaches. So we just paste it in here. Then next one, we will look at the linker. What kind of linker is it? Then let's look at I'm gonna go quicker a bit here. What's the payload? And by the way, if this section is boring for you, you can just skip to the result, basically. But we can look here, what's the origin of the payload? Is it site-specific conjugate? Is there a site-specific conjugation method? What type of conjugation or what method is used? Which generation? Is it a first generation, second generation, third generation, and so on? And these are all things I don't need to know anything about that. I just ask ChatGPT in our platform. You can also do it similarly outside our platform. In our platform, if you look at the most recent publication on the topic, you will come up with some more innovative or specific ones that could be interesting. So what kind of, does it require internalization to release the payload? Is it designed to overcome drug resistance? What is the antibody format? And apologies if you hear the sound, it's my dog who's drinking here just next to me. Is the antibody human or humanized? And then the last questions. Does the patent cover manufacturing or formulation aspects? Yes or no? What's the development stage? Is it preclinical, IND enabling, phase 1 or later? And then who is the applicant? Is it a pharma? Is it a CDMO, a biotech, academic? All right, so we have our 20 questions. That's the max for now. We can always increase that. And you can see here, we look through the organizations, the abstract, the title, the date, and the claims. Let's click on Show Preview. So now it's analyzing the first 5 filed patents in the list. So the ones that are from the last 2.5 years. And it will answer the question. So the first one, well, let's maybe take the third one here. So we see it's an ADC format innovation, dual payload, the target, what diseases, not tumor specific target, platform, payload name. We see the type of the class. We see the type of linker, the payload origin, no site-specific conjugation method, other or not specified, third generation, and so on. Antibody format, the antibody type, no preclinical, and it's a biotech. You can see here what is the company. We see the whole claims here, and let's say we're happy with that. We don't want to adjust our question, which is possible. We want to analyze all. We click here, or we just click on download, and now you will see here these 1,586 patents filed, the most recent filed patents in key regions like Europe, US, WIPO, Japan. This will now be interrogated with these 20 questions, and already 100 are done by now. So this will take probably a minute. You can skip the section, or I can explain how it works. So what we do is we use the best model for this kind of analysis, the GPT 4.1 model of OpenAI. We send these patents one by one with the 20 questions. So it can analyze them. It doesn't know it's you as customer or user asking these questions. It only knows AcademicLabs is asking this question for this specific patent documents. And we don't know, of course, what your questions are. So in this way, it stays secure, private, and confidential. So what will happen? We get for each patent document, it provides the 20 answers, and it works really good against hallucination. Why? Because we only look at the one document. So it's only that one document that is analyzed for each of these specific questions. So it's much less hallucinating than if it would have to create a report based on searching and analyzing information from a lot of different documents. So that's a very powerful way to, in a highly accurate and comprehensive way, analyze a broader set of literature, patents, grants, clinical trials, news, and so on. So once this is ready, it will create an export file, an Excel, and then you can basically use this approach to filter for all these specific criteria. I can focus where it's a certain type of novelty. I could focus on all those targeting HER2. I can focus on all those with a certain disease like lung cancer, or certain site-specific conjugation methods, or in only the preclinical one. So I can play with these criteria to filter down. So now it's basically creating this export file, which can take a bit longer because it's a huge file, of course, with a lot of data. So we basically have the normal export of patents, but then also with 20 additional columns, the AI answer columns. Voila, here we go. Let's open it up. So we see the 20 questions that we have here. So here we can see what are the questions we asked. We saw the query, we saw the filtering, the sorting, the date, and we can go back to the search for repeatability. And voila, here we have our AI answer columns, the yellow ones, and then we see the title of the patent, the inventors, the applicants, the abstract, the link back to the document, and so on. So here I could basically look into, for example, is site-specific conjugation mentioned? Yes. Then we retain 269 of these records. We can look into the specifics here. We can click on any of those. let's say we do everywhere where it's click chemistry. Then we have those. We can look into only it's all third generation, so that's good. Let's say we want to look into more details where it's only, it's about novelty. Sorry, where is it? The novelty is about the conjugation method, formulation. And then we have those, and then we can look further into those specific details. We can go to the right to see any answers here. And we can go to now the specific patent, and we see here who are those players. So that is an approach how you can easily filter down the results. And basically you create your custom filters to analyze those 1,500 patents, which have these lengthy claim texts. But basically in 2-3 minutes, of course I prepared the questions, but that all in itself took maybe 10 minutes. You can just ask like, what are the 20 most relevant questions to ask to learn more or to analyze as a scout patents on ADCs. And voila, it came with like 20 relevant questions, and I went a bit deeper on a few of them to give me some options of answers. So that worked extremely well. And we can do the same for publications to analyze preclinical publications for novel assets, for example. We can analyze clinical trials to identify also biomarkers and other data points, or grants to uncover breakthrough new technologies in development, new drugs in development, and so on. So yeah, I wanted to quickly highlight this option. It basically is extremely fast, and it makes everyone an expert in a topic. So you can just analyze whatever topic you want in record time by offering all these custom filters and analytical options. If you want to try it out, we're happy to give you a free trial, allow you to analyze 500 results for free. All you need to do is sign up on our website and you can get started. And if you need any help, I'm here to help via the chat. All right. I hope this helped, and look forward to hearing from you, how this can help you. All the best. Bye-bye.